5.3 million Americans currently suffer from Alzheimer's disease. Though a cure has not yet been discovered, much research has been performed on the enzyme thought to be responsible for it. My name is Emily Ferguson.
And I'm Hannah Kass.
And this is Beta-secretase 1. So, what is Alzheimer's disease? It is a neurodegenerative disease characterized by the formation and accumulation of plaques and tangles in the brain. Mild to moderate symptoms include memory loss and cognitive difficulty, whereas more severe symptoms include increased formation of plaques and tangles and ultimate atrophy of the brain. Currently, there is no cure, but beta-secretase 1 has been identified to play a key role in the initiation and progression of the disease. So now Hannah's going to talk a little bit about the structure of the enzyme.
Okay, thanks, Emily. So beta-secretase 1 background, it's a type 1 aspartyl protease. Type 1 meaning it's a single-pass transmembrane protein, and protease being, once again, a cleavage protein. Its primary isoform has 501 amino acids. There are altogether 6 isoforms, and those come from differential cleavages of mRNA transcripts that then become translated into the different BACE-1s. Certain active site residues have been identified, so those are residues 93 to 96, also 289 to 292, and they contain primarily aspartate, serine, glutamine, and threonine. It's also notable that BACE-1 acts primarily in different cellular compartments, such as lysosomes and Golgi body compartments, where the pH is 4.5. So taking a look at the structure, the tertiary structure, you can note that it's comprised of a fair number of both alpha helices and beta sheets. There are also 3 different domains. There's the larger extracellular domain, and this is where that catalytic activity comes into play. It has a helical transmembrane domain that's fairly small that keeps it attached to the membrane, and as well as a small cytoplasmic domain whose function has not yet been identified. And so now for the the role of APP, or the role of BACE, it cleaves APP, which is amyloid precursor protein. It's also that type 1 integral single transmembrane glycoprotein, meaning it goes once through the membrane. Also, it's a glycoprotein, so it does have that glucose attached to it. And APP695 is the major isoform of amyloid precursor protein that's expressed in neurons, and so that's what we're going to be talking about for the remainder of our presentation.
Yeah, so now that Hannah's touched on the structures of the proteins we're dealing with, I'll go into the cleavage of APP by beta-secretase 1. APP can actually be cleaved by in 2 distinct pathways. The first of which is a non-amyloidogenic pathway, and that's when it's cleaved by alpha-secretase. So when alpha-secretase cleaves APP, you get a secreted APP alpha that is released into the extracellular fluid, and that is actually normal for healthy brain activity. However, the amyloidogenic pathway involves beta-secretase. So when beta-secretase cleaves APP, you get a similar secreted APP beta protein that's released. But the more important aspect is that once it's cleaved, what's left in the membrane is known as a C-terminal membrane-bound fragment. And when it's cleaved by beta-secretase, that fragment is about 99 amino acids long. That fragment that is left in the membrane becomes further cleaved by gamma-secretase, and what that results in is the release of amyloid beta protein, which is a very important protein. Amyloid beta protein is actually present in small amounts in humans, in healthy adults, but in that way there is small soluble monomers in cerebrospinal fluid and blood. However, in the brains of Alzheimer's patients, you have an accumulation and increase of the amyloid beta proteins, and that's what ultimately leads to the plaques, which are insoluble in the brain, which is what causes the disruption of normal brain activity in Alzheimer's disease. So just to summarize that last cleavage, the cleavage of APP by beta-secretase, beta-secretase cleaves, the secreted APP beta is released into the extracellular fluid. You're left with your C99 membrane-bound fragment, which is further cleaved by gamma-secretase to release amyloid beta protein into the extracellular fluid, which ultimately leads to Alzheimer's disease. So we were unable to find a specific mechanism for the cleavage of APP by beta-secretase, so we would like to propose one. And we'll be proposing this based on our knowledge of the active site of beta-secretase. We'll be using the one that's the amino acids 93 to 96. Also recall that this is occurring in a pH of 4.5, which will be important in this mechanism. And then some new information, the cleavage point that we'll be focusing on is the cleavage between methionine 671 of APP and aspartic acid 672 of APP. So about the mechanism, so as Hannah talked about at the beginning, the active site includes both a serine and an aspartic acid. But although serine, the OH, would be a stronger nucleophile if it were able to be deprotonated, because we're in a pH of 4.5 here, it would be difficult to deprotonate that, so we chose aspartic acid as the major player of the active site and playing into the cleavage of APP by beta-secretase. So what actually happens? We propose that the aspartic acid nucleophilically attacks the carbonyl group there, and at the same time, the amine group here gets protonated by water. Remember that we are in an acidic solution. Then what happens there is that this now negatively charged oxygen gives the lone pair back to the carbon, which ultimately breaks the peptide bond, and that's the actual cleavage that's occurring. So once that cleavage occurs, you've got your aspartate 1 for your amyloid beta protein, and now all that needs to happen is the enzyme needs to regenerate. So the OH from the protonation of the amino group that's left over will come in to attack that carbonyl group a second time, which will ultimately break the bond between the active site aspartic acid and the methionine 671. So what you're left over with is your regenerated enzyme, so it can continue doing its function. You're left with the C-terminal end of your secreted APP beta, which goes into the extracellular fluid, and you're left with your N-terminus of your amyloid beta protein, which in this case begins with aspartic acid 1. So that's our proposed mechanism for the cleavage of APP by beta-secretase 1.
And now we're going to move into our discussion of our primary literature paper. So we looked at amyloid beta protein, GLUT11 is the major beta-secretase site of BACE-1, and shifting the cleavage site of alpha-beta, aspartic acid 1, contributes to Alzheimer's pathogenesis.
Okay, so I'm just going to give a little introduction to the paper. There is a lot going on in this picture, but there's a few things I want everyone to focus on. So when we proposed our mechanism, we said that the cleavage site occurred at the N-terminus site of that aspartate 672, or the aspartate 1 of amyloid beta protein. So, but there are actually two possible cleavage points of beta-secretase 1 on APP. The second of which is at a glutamate 11 site right here. So this part of the picture is showing that when alpha-secretase cleaves APP, you're left with this C83 membrane-bound fragment, which will be important in the later discussion of the paper. However, when beta-secretase cleaves it, when it cleaves at the aspartate 1 site, you're left with that C99 membrane-bound fragment. However, when it cleaves at the glutamate site, you're left with a C89 fragment left in the membrane. So those three fragments will be important for the discussion of the paper. And what the paper is really trying to do is use this background information to determine the physiological consequences of each cleavage point by beta-secretase 1.
Okay, methods. So the first thing that they had to do was create a creation of a cell line, so different cell lines that would express different either APP or BACE-1 or both or neither, to kind of get at the differences between each of the different proteins and the roles they play. And then they took those cell lines and they did an immunoblotting or Western blot analysis on them. So they ran all the proteins through an SDS page gel and then transferred those proteins to a membrane where they could then be tagged with antibodies and identified. And finally, they did an ELISA analysis on the amount of, to kind of quantify the amount of amyloid beta protein that was in each of the cells. For the generation of the cell lines, they started out with the HEK cell, so that has no amyloid beta protein, or no amyloid precursor protein, I'm sorry. So no APP and no BACE-1, but we'll note then all of these beta-actin is constitutively expressed, so we know kind of that they're all being expressed at the same levels, each of the cell lines. So the first thing that we had was they took wild type APP, for example, the APP that might be expressed in our cells, and they created a cell line called HAW with that. They then took a second type of APP, a mutated APP, that has been shown to increase, to have increased prevalences of Alzheimer's disease in populations that express this mutation. They called this the Swedish mutation APP, and cells that contain that were called 20E2 cells. Then they took that wild type APP again, and they had a cell that expressed both APP and BACE-1. And then they took the Swedish mutation and had a cell that expressed both BACE-1 and APP. So one of the major findings that they had was that amyloid beta GLUT1 primary cleavage site in wild type. So what that means is they looked at each of these different cell lines. So when just APP wild type was expressed, you're getting that C83 fragment, you're getting that normal cleavage by alpha secretase. When you take into account wild type alpha beta protein or amyloid precursor protein, as well as BACE-1, you're getting that C89 fragment. So that GLUT11 cleavage site on APP is what is being cleaved by that BACE-1, compared to, for example, when you have the 2BE2, the Swedish type, and BACE-1, you're getting that C99 fragment, so that aspartate-1 cleavage site. The next thing that they looked at was just in general how BACE-1 expression induces that beta-secretase pathway. So once again, when you take those cell lines, HAW and 20E2, where no base is being expressed, you're getting that normal alpha secretase cleavage, that C83 fragment. When you're expressing beta secretase with the BAW cell lines and the 2EB2 cell lines, you're getting those two different fragments showing that we're going down that beta secretase pathway, that beta secretase is doing that cleavage because we're getting that C89 fragment and that C99 fragment. And then finally, kind of the takeaway from all of this is that we increased amyloid beta production by the upregulation of BACE-1 and the shifting of that prime cleavage site from glutamate 11 to aspartate 11. So we see that because if we compare cells in which just APP was being expressed versus BACE-1 and APP being expressed, we find a higher level of that amyloid beta protein that's being expressed. Similarly, when we take that Swedish mutation and the 2EB2, which had both the Swedish type and BACE-1 being expressed, we get that higher level of amyloid beta protein being expressed. And then when we look at wild type APP being expressed versus Swedish type APP being expressed, take a second to remember that Swedish type APP that's cleaved at the aspartate 1 site rather than the GLUT11 site is the one that has a higher incidences in Alzheimer's disease. When we take the Swedish type, that aspartate 1, compared to the GLUT11, we find a higher level of amyloid beta protein accumulation in cells that are being cleaved at the aspartate 1 site.
So in conclusion of everything, that paper that Hannah just summarized for us, the results of that really showed that beta-secretase 1 does play a huge role in the accumulation of amyloid beta protein, and that's really big. And we also know that the cleavage site aspartate 1 is really the major player in the incidences of Alzheimer's disease. So much research has been performed on these specific things. We know so many specifics. We propose a mechanism. There's so much that has been done, but there's still no cure for Alzheimer's disease. So what we would like to end with is that further research in Alzheimer's in general needs to be performed, and research on beta-secretase 1 should be continued, as it might play a really vital role in finding the cure for Alzheimer's disease. So thank you, and here are our references.